Hi, so good morning, everybody. Yes, I'm a postdoctoral associate here in psychiatry, and before the rule changed that small grad recipients had to be faculty members, I had the opportunity to apply for a CFAR small grant and receive one while I was on a T32 fellowship through the Interdisciplinary Research Training Program in AIDS here at Duke. And so that's what I'd like to tell you about today. First, I'm just going to run through generally some background about the project, just to give you some context of why we thought this was an important study to do. And then, of course, I'll run through our scientific approach as well as our results and then conclusions and future directions. So, in 2007, the diagnostic criteria for HIV-associated neurocognitive disorders, or HAND, was revised into these three categories. And it was really done to account for sort of the changes in the manifestation of neurological complications of HIV that were resulting from sort of the modern age of highly active antiretroviral therapies. Really, HIV-associated dementia was becoming far less common, and becoming far more common were more mild neurocognitive impairments. And so those first two categories were created to sort of account for these new manifestations of HAND. And the Charter study, which is a large multi-site trial here in the United States, found that about half of individuals living with HIV have some form of HAND, so some kind of neurocognitive disorder. And obviously the risk of HAND increases along with HIV disease progression, but most cases these days do fall into those mild categories. Now, we know that HIV is a brain disease, right? So while it doesn't infect neurons directly, within days of infection, it does infiltrate the blood-brain barrier, and through indirect mechanisms, it does cause neuronal damage. And so we've seen that in MRI studies, that there's damage to gray and white matter in the brain. And of course, we think that's what leads to some of the neurocognitive impairments that we observe. Now, neurocognitive impairments have been consistently linked with both increased HIV risk behaviors and suboptimal medication adherence, both of which clinically are very concerning things that we would want to address with an intervention. So that got us thinking about what type of intervention could we use to attempt to address these neurocognitive impairments that might help address some of those sequelae, so like risk behaviors and suboptimal medication adherence. So we started thinking about working memory as a possible therapeutic target. So working memory is a core cognitive function. It involves the temporary storage and manipulation of information from our current environment that we use to somehow perform complex cognitive tasks. So clearly, working memory is linked to decision-making. Impaired working memory is linked to impaired decision-making, which means it could be mechanistically an important thing to address in terms of these neurocognitive impairments I was just discussing. One theory posits that suboptimal decision-making results because there's these two decision systems in the brain that get out of balance. So there's the impulsive system, and then there's the executive control system. And so our thinking was, if we could strengthen the executive control system by strengthening working memory, maybe that would lead to some positive results for these impairments. So that's what led to the CFAR small grant that I applied for. We called it Project Impact. And our idea was to use cognitive training to address these impairments. Cognitive training is essentially performing exercises that are specifically designed to enhance cognitive skills. And in some cases, it can also involve teaching a person compensatory strategies to deal with whatever deficits they're experiencing. But we were more focused on the exercises part of cognitive training. And what cognitive training does is it really maximizes learning through individualized progression. So as people are going through a training program, things get increasingly more difficult the better they perform, so that over time their skills are gradually enhanced. It can be delivered via a therapist. It can also be done just by a workbook, but what we were most particularly interested in was computerized cognitive training, because that would be highly cost-effective to deliver in clinical settings. So the aim of this project was to test the feasibility and the preliminary efficacy of a computerized working memory training program on persons living with HIV. All right. So in terms of our eligibility criteria, we tried to enroll pretty broadly with not a whole lot of restrictions. These are the principal eligibility criteria. So we recruited HIV-positive individuals, ages 18 to 65. Because of the complications, the confounds that drug use introduces, we required them to not have an active substance use disorder or be actively using illicit substances. And then we also, during the screening visit, we tested their working memory and required that they demonstrated at least some degree of working memory impairment to be able to enroll in the trial, because we wanted to be able to see an improvement. So if their working memory was amazing, obviously they weren't going to improve much with our trial. We did assessments at a baseline visit, and then we did a follow-up assessment at a post-visit right after training was completed to evaluate intervention effects. And for our visits, we included a full neuropsychological battery that included working memory testing. But then we also assessed other domains of function because we wanted to examine the specificity of the intervention. So is working memory training good just for working memory, or does it perhaps extend to other domains of function? The design was a randomized clinical trial, and we used a commercially available program called PSS Cog Rehab. That's what we used for the training. It was developed by a neurologist and has been shown to be effective in the literature, so it seemed like a good option. And we asked people to complete 12 sessions over 6 weeks. The sessions were about an hour long, and we allowed as long as 10 weeks for people to complete sessions to account for canceled appointments or missed visits, because we knew for some people they might not be able to come twice a week every week. There were 2 conditions, and people were randomized to one of those 2 conditions at their baseline visit. The first condition was active cognitive training, or ACT. And in that condition, participants completed 5 modules from the training program that were specifically designed to enhance working memory. So it went through that individualized progression over time to build their working memory skills. In the control condition, it was an attention-matched control. So they also came in for 12 sessions. They completed the same 5 modules, but we restricted the modules so that they would not progress in difficulty. So participants got the easiest item every time at every session, which was admittedly a little boring potentially, but it didn't challenge their working memory and therefore theoretically wouldn't enhance their working memory. So this is a screenshot of what the program looks like. And this is an example of one of the tests that people got. So this is essentially a digit span module that people would complete to try to build up their digit span over time. So the computer would audibly pronounce a number and then also show it on the screen. And then the screen would blank and participants would punch in the numbers that they heard in order. For the ACT condition, when they got one right, they would do another one, and then the number would go up. They'd be asked to do 4 numbers, and then 5, and so on. And if they started getting some wrong, it would backtrack to the prior set. So it was sort of accommodating how the person's performance was. And over time, people got farther and farther along in the module. For the con condition, they just got the 3 numbers every time. They never got more digits than that. Okay. So we screened a total of 33 people for the study. What we first did was a brief preliminary screener where we kind of told people about the study and said, you know, are you even interested in doing this? And so a lot of people screened out even before being scheduled for the in-person session. So we scheduled 33 people for the in-person screening session, and 12 people were not eligible. Mostly, the most frequent reason was because they didn't have any impairment in their memory function, which is great, but it excluded them from the study. And then there were a variety of other reasons that people were excluded. So that led to us randomizing a total of 21 people. We randomized 11 to the ACT condition and 10 to the control condition. And our retention rates were pretty good. Everyone in ACT completed the post-training assessment, and then we lost 1 person in the con condition. So 1 person was lost to follow-up. In terms of participant characteristics, the main thing I want to point out on this slide is that there were no differences between the 2 groups, at least statistically speaking, on any of these characteristics. So demographically, the samples were similar. In terms of HIV disease characteristics, the samples were similar. They, on average, were prescribed 2 ART medications, and all of them were currently on antiretroviral therapy. In the ACT group, everybody completed all 12 sessions, which was great. In the CON group, not everybody completed all 12 sessions, which was what I was alluding to a little bit when I said it could potentially be boring. I'm suspecting that maybe they weren't particularly motivated to always attend their session, maybe. But 2 people didn't complete all 12 sessions, and one of them was that person that was lost to follow-up. At the post-training assessment, we had people complete an intervention process assessment where they could give us feedback about the intervention and let us know what their thoughts were, what their experiences were with completing it. Overall, people rated it really positively. People said they were overall satisfied with the intervention. I didn't put it on here, but the helpfulness ratings were similar to the satisfaction ratings. So people were satisfied with it. They found it helpful. They said that they enjoyed the sessions. They didn't particularly find it difficult to use the computer, which were all things we were concerned about in terms of feasibility. So we were really happy to see what these results were. In terms of perceived benefit, for the most part, the ACT group more strongly agreed with statements about the benefit of the program. So while some of these weren't quite statistically significant, in general, the ACT ratings were a little bit higher, making us think that maybe the ACT people were picking up that something was changing for them. People rated the program okay. We asked if they thought the program was high quality. They were like sort of neutral, kind of agreeing with that statement. So they didn't love the program. And then they also weren't sure about the number of sessions being helpful. Again, they said, well, some of them were neutral about it. Some people said maybe more sessions would be helpful. But overall, these results made us feel really good about the acceptability of this as a possible intervention. So to look at intervention effects over time, we ran a series of repeated measures and COVAs, looking for a time by condition effect. And this is the effect for working memory. There is a significant interaction effect there. So what's shown on the y-axis is people's T-scores. For the neuropsychological test that we use, the T-score is basically the person's normed performance. It's a standardized score. 50 is considered the mean, and then 10 points below that would be 1 standard deviation. So you can see in blue that at the start, both groups were below, sort of in the mid-40, low 40 range. For the ACT group, at the post-training, their scores had actually gone to just above 50, meaning they had returned right to normal levels of working memory function. And then in the CON group, their scores were basically largely unchanged. Like they went down just a little bit, but basically we would say that wasn't a meaningful change in the scores. So this is kind of exactly what we were hoping to see. So I was thrilled when I ran this and saw this pop up on the screen. And then what's interesting is in all other domains, there were 0 effects. No time effects, no group effects, no interaction effects. So the effect was very specific to working memory. As with any study, there were challenges here. The number one for us was recruitment. Getting access to patients, structural barriers at the clinic made it so that recruitment just went really, really slowly for us. We recruited for 6 months longer than we planned, and we still were well short of the sample size we were hoping for. And part of that was that some participants, as I mentioned at the pre-screening, would say they weren't interested in doing the study because it was just too much of a time commitment. So having to come into the lab twice a week and do all these sessions for people who were working or who had other obligations, it just wasn't practical. But the other thing that we did was to try to limit attrition. We excluded people who lived more than 15 miles away. And so that ruled out some people too who might have been willing to participate. So thinking about those challenges, there's some changes that we think would warrant the next time that we try to do this, what we would want to do. So one, switching to a web-based intervention would be ideal, because then people can complete sessions at home, right? They can come into the lab if they want, but they also have the flexibility of just doing it at home. We also used a pretty minimal dose here. 12 sessions is just not a lot of time. So increasing both the frequency and the duration of the intervention would be important next steps. And also switching to a more engaging interface. Ultimately, I do think the participants found the program that we used a little bit boring. And other things are also, expanding, so we're not excluding to just those 15 miles, using a longer follow-up period, and then potentially also looking at other specifically high-risk groups, such as stimulant users. So in conclusion, we found exactly what we were hoping to find. It seems feasible, it seems acceptable, the results were really promising in terms of the training's ability to improve working memory. Our conclusion is that cognitive training seems like a really promising intervention. And I'm happy to report that within the last couple months, we actually received a notice of grant award for an R21 study to do the next phase of this project. And we would have never gotten this study without the pilot results. So I'm just really appreciative that we were able to do this pilot study because we're really excited about this next phase of the project. For this next phase, we're doing exactly all the same things I just told you that we think we should do. We're going to do more sessions, we're going to switch to web-based, and we're going to add a 1-month follow-up period, and we're going to focus on stimulant users because we think that's a special population that really needs attention, and particularly because of HIV risk behaviors, really needs an intervention like this. We're anticipating that recruitment will start in January, and we've partnered with the Human Cognition Project at Lumosity to use their web-based training program. So we're really excited about this next step. So lastly, I just want to acknowledge my lab, everybody at the HIV and Addictions Research Program, and particularly my faculty mentor, Dr. Christina Mead, and then also particularly Pooja Patel. She was the clinical trials assistant who ran all the training sessions, and there's no way this study would have been done without her, so I really appreciate her help. Thank you.
Do we have any questions? Sheri?
Answered all of them, right?
How do you assess working memory at the end? How do you assess working memory at the end?
So we used a neuropsychological battery that included digit span forward and backward, and then also the paced auditory serial addition test. So it was a fairly short battery to look at working memory. And one thing that we're going to do in the follow-up R21 is we're going to actually extend that battery and add a couple more tests so that we can get a more comprehensive picture of working memory because the battery was a little bit limited.
Thank you.
Yeah.
Disorders versus an acute, they might have a lesser effect.
Sorry, I couldn't quite hear you. Are you asking about acute infection versus?
No, so are your subjects chronic or acute?
Oh, I see. Yeah. So that was actually an exclusion criteria that they couldn't be acutely infected with HIV. Yeah. Sorry, we had a handful of exclusions, and just for time, I didn't run through all of them, but that was one, because we would suspect there to be differences.
So a common criticism of brain training is that it doesn't transfer. So have you tried looking at that in particular?
We haven't really looked at that yet. So that's definitely something that we're hoping to do. This was like a simple, basic efficacy trial. So that's definitely something we'll pay attention to, because you're right, there has been some criticism of cognitive training, particularly because of that. Yeah.
I think we need to move on.
Okay. Thank you.